Yes.
Atif, Minashi, was it? Yes, okay. We discuss till meiosis, mitosis, correct? Then asexual mode. We were to start vegetative propagation. Oh, we started vegetative. Okay, we did the natural and natural vegetative propagation. We have to start with artificial, correct?
Yes.
Okay. No doubts till now? No doubt in for anyone in this batch? Okay, so what we'll do is, like, before we begin every new class, we'll brush up certain concepts from the previous one, so we all are on the same page. The next set of learnings, okay? Okay, and I have received Atif's assignment. Mazia and Minashi, you have not submitted your assignments yet. You will be, right? You should. You have received the assignments, correct, Mazia and Minashi?
Yes, sir.
Yes, so please submit it so that I can see how well you are progressing. Okay, everyone. So let me ask Atif. Atif, what did you understand from asexual reproduction? Just shed some light on asexual reproduction, various modes.
Yes, in asexual reproduction, there is only one sex involved, and there are 5 types: budding, fragmentation, regeneration, vegetative propagation, and binary fission.
Yes.
Yes.
There can also be multiple fission, but basically it comes in the same category as fission. Okay. Okay, cool.
Okay.
And what did we understand? So I told you the difference between fragmentation, regeneration, and budding, and why, which is the most natural mode. Atif, which is the most preferred natural mode for reproduction, asexual reproduction in Hydra? What do you think? Budding. Why?
Because budding can be done on the organism's own accord. The other one require external factors.
At will, it can do that. Right. Perfect. And how is regeneration different from fragmentation, Minashi? You remember how regeneration is different from fragmentation?
Fragmentation means the parent body divides and it forms several fragments. And regeneration means it's the ability of growing back.
Going back what?
Like the lost part of body.
But I also told you it's also the ability to grow back the whole organism from the lost body part. Remember both the things I said. In Hydra, regeneration is a type of asexual reproduction. But in a lizard or in a human, it is not a type of reproduction. Remember, Minashi? Can you tell me why? Why in Hydra it's a type of regeneration? Because the whole organism can be regenerated back from any lost part, right?
Yes.
So what is the major theme, major difference between the two? If you have to summarize in one sentence, what is the major difference between the two?
Regeneration can be considered as a true mode of reproduction. For lizard, it's not a reproductive. I mean, it's a type of reproduction.
Fragmentation. Atif,
Yes.
do you agree with this, Atif? Would you like to add something?
I'm not really, I didn't really understand what she said.
He said reproduction is not, regeneration is or is not a true mode of reproduction. Minashi, what did you say?
It's not.
Regeneration is not.
Yes.
Yes, so she said regeneration is not a true mode of reproduction, whereas fragmentation is. Do you agree?
Yes.
Yes, because for regeneration, you need an organism that can regenerate the whole organism back from the lost body part, which happens only in few organisms. And also for regeneration or fragmentation both to happen, you need an external factor that can in the first place split or fragment the organisms into different parts, correct? Very good. We started with vegetative propagation. Okay, Mazia, what do you understand by vegetative propagation?
Um,
Yes, you're speaking, Mazia. What do you understand by vegetative propagation? Just tell me the basics from the last class. Mazia, I'm not able to hear you. Can anyone hear Mazia, Atif or Minashi?
No.
Yeah, you're not audible. Yes, now you are mute. Please unmute and try to speak. Let's see. Yes, Mazia. I can hear some voice, some not your voice. Are you having issues? You can just write text to me if you're having issues with the microphone. See if it can get sorted. So let me give you a quick recap for vegetative propagation since Mazia is not able to speak up. So vegetative propagation is basically it happens in higher plants through which plants without going through germination or sexual reproduction can just propagate and form more plants. It is a faster mode, because you do not need the plant to grow from the seed and become mature. You can just take any matured part of the plant and the plant can start growing from there onwards. Okay. Now, vegetative propagation are of two types, natural occurring and artificial ones that we do. You all can see the screen that I'm sharing. The screen is visible, right? Yes. So in naturally it can happen through roots, stems, leaves, and bulbs. And it's clear, root, stem, leaves, and bulbs, no doubt in this. Today we have to start with the artificial ones. Basically, this is the types of vegetative propagation that we carry out. Did I give you the definition for artificial one? Should I give it again?
Yes, sir.
Yes. And I also asked you to go back and read about differentiation, dedifferentiation, and redifferentiation. Did you all do this assignment? Self-learning thing? Did you revise? Yes, Atif did. What about Minashi earlier? You did, Minashi?
Yes, sir.
Perfect. Fazia, able to speak? You're writing. Okay, you also did. Good. So we'll be discussing this in a bit. Let's first go to the different types. So cutting, layering, grafting, and tissue culture. Okay. Let's start with the first one for this class, which is cutting. Okay, so let's start the discussion. What do you understand by cutting? And what is cutting in vegetative propagation? See, cutting means we are cutting something, right? What are we cutting?
Stem of the
Stem, right?
yes.
Yes. It can be stem or it can also be leaves. Okay. So let's define it. Write down. In this process, a part of the plant, specifically stem or leaves, a part of the plant, specifically stem or leaves, is cut. So you simply have to just cut it and plant it in the soil. Are cut and planted in the soil.
Can you repeat from the
Okay, that's I said in this process a part of the plant, specifically stem or leaves. A part of the plant, specifically stem or leaf, is cut and planted in the soil. Is it clear?
Yes.
The new plant is formed from the new plant, continue in this only. The new plant is formed from the adventitious roots. So it's very clear, right? If we are just cutting a stem or a leaf and putting it in the soil, what it does not have, it does not have a root. So for anything to develop in soil needs to have roots, correct? Right? So roots, and I also told you that roots, if developed in a place where it should not have been there, it's called adventitious. So stems or leaves, when you cut and put it in the soil, adventitious roots starts growing, and then a plant starts growing out of it. Is it clear, everyone? So write down, the new plant is formed from the adventitious roots developing from the cut part. Developing from the cut part.
Can you repeat the second point?
The new plant is formed from the adventitious roots developing from the cut plant. You understood this term or this sentence, Atif? Clear?
Yes.
Yes. Now sometimes we have to induce this process. Now this process naturally will not happen. That's why this particular type does not comes in natural mode of propagation. So sometimes we have to artificially induce roots in these cut parts. And how can we artificially induce roots in any plant part? Class 11th, remember plant hormones? Plant growth hormones, class 11th?
Yes.
Plant growth regulators used to call them, right? So write down, sometimes the cut parts are treated. Sometimes the cut part are treated with plant hormones. With plant hormones to regulate or induce root formation. Is it clear, everyone? Any example of cutting that you are aware of? Rose.
Rose
The
plant.
sugarcane?
Yes, sugarcane. Rose and sugarcane. There are many more plants. There's a list. So you have to make a list of at least 5 most common and starts looking into books. Whenever you find a plant, see if it can be grown through cutting, grafting, or layering. You should know that. So rose and sugarcane you can write as examples in exams. Okay. Now the next one is grafting. What do you understand by the term grafting? I've already told you about grafting, right? Putting something, taking something out and putting it somewhere else, like a tissue graft, skin graft that we do. We call it plastic surgeries. Remember this example, everyone? Yes or no?
Yes.
Yes. So grafting. Now, for grafting, we have to cut, correct? Now, can anyone tell me how is grafting different from cutting then? How do you think is grafting different from cutting? Please. Cutting is also we are cutting a part and we are putting it in the soil. Then what makes grafting different as a procedure?
Sir, I'm not sure, but yeah, we are cutting a part of a plant and putting it in the soil, and then we are cutting a part from another plant and then putting it on top of like the plant that we originally cut. I'm not sure, like combine them together.
Yes, you're right. You're right. So in one procedure where it is cutting, we are cutting a part and putting it directly in the soil. Correct? Whereas in this part, we are not putting it in soil. We are putting it at another plant. So it's like grafting a tissue from one place to another. You understand?
Yes.
Now this is like, let's suppose there were two plants, and I have discussed this, I think, above also. What's joining in? It's joining late. Let Mama join. Okay. Good evening, Mohammed. You're there, Mohammed? Good evening. We are discussing vegetative propagation. We have discussed cutting. You joined a little late. We are discussing grafting right now. Okay. If you are,
Yes, sir.
yes,
Good
welcome.
evening, sir.
We're a little late, but we just discussed cutting. And just for you, I would like Mazia to repeat. Mazia, can you just tell Mohammed about cutting, what we discussed about cutting, whatever you have written?
Yes. Um,
Mazia, you are not audible if you are speaking. Minashi, can you step up?
Yes, sir.
Yes, Minashi, can you tell what we discussed about cutting right now? Just
In
yeah.
cutting, the part of plant, especially the stem or leaf, is cutted and planted in the soil.
Yes.
Then the new plant is formed from the adventitious root, developing from the cut part.
Yes. Is it clear, Mohammed?
Yeah, okay.
We are
Thank you,
just
sir.
cutting a part of the plant, and specifically there can be two of these parts, stem or leaf, and we are directly putting that cut part in soil to grow. That's cutting. And sometimes we also need to give them plant hormones to induce this root formation. Otherwise, roots will not form very easily. Okay. Rose and sugarcane are examples of it. Okay, Mohammed? We just started with grafting now.
Yes, sir.
Now, grafting is where we are cutting a part of plant, but not putting it in soil, but putting it on another plant, grafting a tissue from one place to another. So let's say there are two plants. One plant, okay, is this. It has a very good flower system, which will eventually give good fruits. And there is another plant which has a very elaborative and very good root system, but the shoot system is not that, it's doing fine. So we cut this part, okay, and we transferred it to the other plant and we grafted it here. Is it clear, everyone? Now when we are grafting it, what basically we make a oblong cut. Remember we talked about this? So this is the stem. In the stem we will make a cut like this, and then we will take another tissue that we need to graft, which will be like this. Okay, and we will put this here and we'll wrap this whole thing so that there is no infection occurring. Is it clear? Because plant cut parts are also like open wounds as a system, right? They are also living systems, and plants also have their viruses and bacteria that can attack on it. Make sense to all of you? So write down, in this process, in grafting, the cutting from some other plant the cutting from some other plant is attached to the stem of a plant is attached to the stem of a plant that is rooted in the ground. That is rooted in the ground. Is it clear, everyone? Makes sense or no? Tell me.
Sir.
Now, I have a question, okay, and this question is for Muhammad, let's say. When we have the option of cutting, what do you think is the advantage in grafting? Why are we going for grafting? What does grafting has that cutting does not? Muhammad, to understand the concepts. Any idea? Muhammad? Are you there? Just say yes to acknowledge my question that you heard it. I cannot hear you, Mohammed. You're there? If you're speaking, you're mute. I'm able to hear you. Let's go to Atif. Atif, you're there? What do you think? Which one do you think has a better advantage?
Grafting.
Grafting, right? Why? Why do you think so?
Because the grafting can produce plants with better yield. Like the example you gave, if you take a plant with a good root system and a plant with a good shoot system, and we graft them, we can get a plant with like a really good yield. And we can also like create hybrid plants.
Yes, it has both the systems, and also in cutting, the plant have to develop roots altogether from the beginning, right? It has stem and leaves, but it has to develop roots, correct? In grafting,
Yes, sir.
the plant just have to accept the other plant and merge. Both the systems are already in place, a good root system and a good shoot system. So you do not have to develop something from the beginning, but you have to just make a good match, correct? Grafting can be a little tricky to do, but once successfully done, it is better a system. So write down the tissues of the graft. The part that we are putting in is called graft. The tissue of the graft become integrated with the tissues of the rooted plant. And develop as a single hybrid plant, like Atif said. A single hybrid plant. Clear? Everyone has written down? Clear to all of you? No doubts. Say yes or no?
Yes, sir.
Yes. Now, who can give me examples of grafting? Any example of grafting? See, one could be, how many of you have seen anyone making a graft? We often do it for fruit trees, remember? Many fruit trees are propagated through grafting, because once we find a good root system which is very, very productive, we would like fruits to be grown on that system. Okay, so write down tomatoes, avocado is an example. Rose can both be propagated through cutting and also through grafting. Okay. Here you have tomatoes, avocado, rose, Even mango, hybrids of mango is done like this. Is it clear, everyone?
Sir, what about pear?
What about
Pear.
pear? Yes,
Pear fruit.
pear can be grafted. Yes. But for grafting, you have to remember that the tree does not have to be very old, because we need young system which can develop over time, right? So it has to be relatively younger so that the graft can be accepted properly. Now, third one is layering. Grafting and cutting is clear. What do you understand by layering? What is layering?
Sir, I'm not sure. I think you like bend one part of the plant into the soil and then it like grows a separate plant, something like that. I remember it very vaguely.
Yes, true. You're perfectly correct. By layering, we means we are layering it. We are bringing it back to a level where we are again layering it in the ground. Okay. So this happens this can only happen. Now tell me what is the, okay. What is one disadvantage or difficulty in grafting? Tell me. If you are grafting a shoot from one plant into the root system of another, what has to be taken into consideration for grafting? For cutting, we need nothing, just cut and put it in soil, simplest. But when we are putting an open cut part of a plant in soil, it may grow roots, most of the time it does, but it can also get infection from the soil bacteria and viruses, correct? Right? Yes or no?
Yes.
Yes. So grafting gets rid of that infection part because we cover the grafted part properly. But we are putting one part of the plant to another part. Now, what do you think should be taken into consideration? One very important thing that we cannot neglect is that both the plants, the graft and the rooted plant, has to be similar, has to be of similar girth, right?
Yes.
They should have the same diameter of their stem. Don't you think so? If the rooted plant is this and the shoot you cut is this, will it make a good graft?
No.
And if it is vice versa, the rooted plant is this and the shoot is this, will it make a good graft?
No, sir.
No. So their diameter of the stem, the girth, which we call, should match. You can write this point also, that for grafting, the two plants should be compatible in their shape, size, and also anatomy. By anatomy, I mean xylem and phloem. Remember studying xylem, phloem, class 11th, plant physiology?
Yes.
Yes. So if we see a plant like this, and it has xylem bundles running like this, then you would like another plant whose xylem bundles are also running like this, right? Because when they will become a single plant, the attachment will be easier, correct? If there's a difference in this positioning anatomy of these bundles, then it becomes difficult. So for grafting, it's limited. Not all plants can be grafted on all plants. So only few plants can be grafted with other plants. Is it clear, everyone? So it's a better technique, but it has its own limitations. And you have to remember the limitation and advantage for all the techniques. Clear? Shall I move forward? Yes. Now layering, as Atif said, layering is where we bend a part of a plant and put it back in the soil, from where a new plant emerges. Now it seems very, very simple, but tell me, can we do this for all plants?
No, sir.
Why? Why, Atif, you're right. For which plants we cannot do it? I think it's just logical. Can we do layering for coconut plant, for that matter?
No.
Why? See, what you have to do, you have to take a branch, stretch it enough, with pressure, so that you put it in the soil and then some part of it must be coming out and the rest part you cover with soil. We are not cutting that part. The part is still attached to the plant while it is being lowered and layered back in the soil. Is it clear, everyone? But for a plant which has a very long stem system, suppose this is the ground, it has roots, okay, and it has a long stem, unbranched stem, and then branches start somewhere here, very up on the ground. So can we do layering for this kind of a plant? No, because this branch which has trees, we will not be able to lower it back to the ground. Clear? So for which plants it can be done? The plants which has a shoot system and branches developing very low on the stem, and we call such plants as bushy plants. Remember, bushes have lots of branches arising very low. That's why we use bushes for making boundary walls. Correct? Say yes or no?
Yes, sir.
Yeah. So we need a plant which has a small stem and then there are long branches coming out of that stem. So for this we can do it, because this stem can get lowered and put back in the soil with some part of it outside. Okay, so we put it back here and then we cover it with soil. So this is covered with soil. So the plant is continuous, it is still attached to the mother plant, but over time this region starts growing roots of its own because it's inside the soil, and over time this connection will severed and this will become a new plant. Is it clear, everyone?
Yes, sir.
So layering is, write down, in layering in layering, the stem of a plant, the stem of a plant is bent to the ground, and covered with soil. Covered with soil, with some part of the branch still outside the soil. This part, with some part of the branch still outside the soil. Now next point, the plant part covered with the soil develops. The plant part covered with the soil develops adventitious buds, because we are layering it in the soil. It develops adventitious roots, sorry, not buds. I said buds.
So the plant parts.
What did I say? The part covered with the soil develops adventitious roots. Develops adventitious roots. Okay. And this, and write down the last line. And this attached stem, this part here, this is the stem, attached stem, which is still out. These are the leaves and the new roots that are developed. This part, the attached stem with developing roots, is called a layer, a new layer, like a new plant. That's why this process is called layering, because the new plant, the new developing plant, is called a layer. Clear, everyone? Can anyone give me any example of layering? You know, plants that are developed through layering. Gardeners often do that. Any idea which plants are developed through layering? People, please. Give examples. I already have given you the hint that the plant type has to be bushy so that branches arise very low on stem. So one example could be bonsais. You remember bonsai plants? You know bonsai plants? Who knows bonsai?
I know somewhat.
Yes, Arvind. What do you think is a bonsai? Bonsai.
Isn't bonsai like an art of cutting plants or something like that?
Bonsai are miniature versions of bigger plants. Like you can have a bonsai of a people tree or a banyan tree in your pot. So it's like a tiny miniature living banyan in your pot in the garden. Okay, that's called a bonsai. So because when we are making them, they are modified plants, hybrid plants. We can propagate them through layering because they are very small. Their branches just arise a few inches above the stem, so they can be put back in the soil and you can propagate them through layering. Okay, write down another examples are plants with flexible stem containing plants with flexible stems. For example, flowering plants. All the plants that flowers that are propagated for flowering, they are mostly smaller, bushy, like bougainvillea, rose, and can you give me examples of other flowering plants? Garden flowering plants?
Jasmine.
Jasmine, yes, jasmine also has a very flexible branch. So you understood flowering plants mostly are propagated through layering. Is it clear? Shall I move forward? Doubts? No doubts. Okay. Now, last part is tissue culture. Very important. Now let's come to tissue culture. So what do you understand by the term tissue culture? Anyone would like to answer? Tissue culture. What do we understand by tissue culture? We are taking certain tissues from a plant, of course. You can also do animal tissue culture, that's called, or animal cell culture. When we are doing it for plants, we call it plant tissue culture or plant cell culture. Is it clear, everyone? It started with, like, mostly it's done on plant tissues, but it's also done in animal tissues for research purposes. When it is done in plant tissues, we also do it for propagation purposes. When we do it on animal tissues, we do not do it for propagation. We do it for maintaining that tissue line. We're doing some experiment. You understand? So write down, let's first write down in what is tissue culture, write down. In this process, plant tissues from different parts, plant tissues from different parts are cultured in laboratories. So for tissue culture, you need laboratory. You cannot do it in soil. A laboratory or even a like a petri plate. You need a setup for it. What did you write, Atif? Can you please repeat? Sorry.
In this process, plant tissues from different parts are cultured in laboratory.
Cultured in laboratory to develop new plants. So if we think about tissue culture, doesn't it looks like fragmentation and regeneration? Let's first complete the definition. Cultured in laboratory to develop new plants. Right on next point. This technique, why do you think this technique is helpful? Any idea? It's very helpful to culture plants which are very rare now, which are not found in wild or which are getting extinct. Write down. This technique is helpful in propagating rare and endangered species. Rare and endangered species. Is it clear, everyone? Okay, makes sense. Rare and endangered species which are which are unable to grow under natural conditions, which are unable to grow under natural conditions. So it's simple that plants that cannot grow under natural conditions, so basically we cannot make them grow through layering, cutting, vegetative propagation, or by sexual reproduction, by fertilization, etc. So we do tissue culture on them. And in tissue culture, like I told you, we are using certain tissues from different parts of the plant. We cannot use any tissue from any part of the plant. So what do you think must be the speciality of the tissue which we are using? Anyone? This is where we come to differentiation, dedifferentiation, and redifferentiation. Please, you have to discuss with me. So what do you think should be the characteristic of the tissue that we are using in tissue culture? I said it should be specialized tissue, but specialized in what? Specialized in division, right? In dividing and forming other structures, correct? Yes. Now look carefully, because let's take the discussion further. Let me give you some concepts. Cutting, grafting, layering, and tissue culture. If you look all these methods carefully, isn't it simply fragmentation and regeneration only? Think about it.
The layering is not a form of fragmentation.
Sorry, layering is?
Not a form of fragmentation.
Right. But layering is regeneration. You are regenerating roots from a point where it should not have been. Like, right? So fragmentation is not there, of course, but you are regenerating certain organs or certain structures which were not present there. But now they are present there. Correct? Latif?
Yes, sir.
Yes. What I meant to say is that vegetative propagation is nothing but fragmentation and regeneration, or any one of the two in plants, correct?
Yes.
In tissue culture also, if you see tissue culture, if I tell you that there was a plant, most of the time what we do, we take the tips. So this is a shoot system of a plant. If it is a plant shoot, the tip of the shoot, again class 11th you have studied about the tip, the apical tip has tissues called meristematic.
Meristematic tissue.
Yes, perfect. Meristematic tissues. What is the speciality of meristematic tissues, Arthur?
They are continuously dividing. They help in the growth of the plant.
Yes, they continuously divide. So they have this ability to rapidly divide and form more structures. Now this type of ability, meristematic tissues, can still differentiate, divide. So they have active dividing capability. So the tissue that we choose for tissue culture should be chosen carefully, that it should have active dividing capability. Correct, Adif?
Yes.
Let's talk about the other types of tissues. What happens to other tissues in a plant or in an animal? They stop dividing after a point, right?
Yes, sir.
So here are the three words which I ask you to read: differentiation, de-differentiation, and redifferentiation. So let's try to understand what is differentiation first. What is differentiation? What does the word differentiate means? Question comes, differentiate between X and Y. So what do you do? In an exam, in any subject, if there is a question, differentiate between X and Y, what do you do? You talk about the differences, right?
Yeah.
Yeah. So differentiation is a biological process. When we are talking about differentiation from the perspective of biology, it is a process where tissues or cells divide and take up a role. Okay. So differentiation, tissues divide and become specialized to do something. For example, our blood cells are differentiated form of cells. Like red blood cell is a differentiated form of specialized cell that has a role of carrying oxygen, because on its surface it has a protein called hemoglobin that binds to oxygen, correct?
Yes.
Can the cells of our eyes or our nose or our bones do the same function? No. So different tissues differentiate to become specialized in different functions. Is it clear? That process of maturation of tissues and to take up a specific function is called differentiation. Like some tissues went up to become brain cells. So they start the process of thinking, memory, learning, consciousness. Some tissues went and become the tissues of heart. So their function is to contract and relax and pump the blood, correct? Same is for muscular tissues. Their function is to help the body move, locomotion by contracting and relaxing, correct? So this is differentiation. So let's define differentiation. Differentiation refers to, or differentiation means, a cellular process, biological cellular process, a biological cellular process in which the cell the cell acquires certain modifications. The cell acquires certain modifications in structure and function. In structure and function. Is it clear? So we start from something which is common. We call it stem cell. Have you heard this word before? Stem cell? Stem cell is a common precursor from which we go on forming neurons. Neuron is a special type of differentiated cell which has a specific structure and a specific function. We also go on forming red blood cells, which looks like this, RBCs. We also form muscular cells that are spindle shaped. Muscle cells. We can also form bone cells. Is it clear, everyone? So this process from a common cell line, when we form different structures and functions, is called differentiation, correct? Now each cell is different and unique. Neurons cannot form RBCs, RBCs cannot form bone cells. So which means now these cells have differentiated. Is it clear? Say yes or no? Is the concept clear to all of you?
Yes.
Yes. Now, what do we mean by other two terms? You have to think logically. Dedifferentiation, and then the third one is re. You understood differentiation. Now you have to understand dedifferentiation and redifferentiation. And I will encourage you all to look at the terms and tell me the answer. What does the word D means? What does the word re means? Tell me. Dedifferentiation. What do we mean by deforestation? Mohammed. Deforestation. Quick. So you have to discuss, right? I'm not here to just dictate you things. My motive is to make you understand these concepts, you will never forget them. Differentiation you understood, becoming different, simple. De-differentiation, D means deforestation, demotivation. Defeat. What does the word D means? Yes.
It's like the reduction of something.
Yes, reduction or like removing something, correct? Deforestation means removing the forests. So what will de-differentiation means? Removing the differentiation, correct?
Yes.
Removing the differentiation. Now, what do I mean by removing the differentiation? I mean that from a stem cell, from a common precursor cell, I made many cells with specific structure and function. De-differentiation means losing that ability, losing that special function or losing that special structure and going back to a state from where you can again differentiate. You understand? People.
Yes.
Yes, so write down. De-differentiation means or don't write means, write down. De-differentiation is a cellular process. De-differentiation is a cellular process in which an already differentiated cell, already differentiated cell loses its special structure and function. Loses its special structure and function and gains the ability and gains or acquires the ability to divide and differentiate again. Divide and differentiate again. Is it clear?
Yes, sir.
Yes. Now tell me, what does redifferentiation means? This you have to tell. I talked about two, you have to talk about the last one. What do you understand by redifferentiation? Yes, Arjun.
The differentiation takes place again.
Yes. So re means repeating it, like reproduction, re-examination, redifferentiation. So redifferentiation happens after dedifferentiation. If it is happening for the first time, right from a stem cell for the very first time, it is called differentiation, simple. But if it is happening for the second time, and for it to happen the second time, it must have undergone dedifferentiation. Is it clear? Making sense or not? Yes or no? People, say yes or no?
Yes, sir.
Yes.
Yes.
So to redifferentiate, you have to dedifferentiate. Okay. So differentiation, going back is dedifferentiation, then again coming to differentiated form is called redifferentiation. So write down redifferentiation is the process where the cell divides, the cells divides and produce more cells. The cell divides and produce more cells, which once lost their dividing capacity. Which once lost their dividing capacity and regained it through dedifferentiation. This one. And regained it through dedifferentiation. Is it clear, everyone? Can you repeat what I just said? Can someone repeat this last definition of redifferentiation? Tell me redifferentiation, people. Anyone? Just repeat redifferentiation so I can figure out. Yes, Atif.
Redifferentiation is the process where a cell divides and produces more cells, which once lost their ability to divide and regain it through dedifferentiation.
Yes. So they regain that ability through dedifferentiation and then they redifferentiate, again divided. Okay. Very good. So which type of tissue do you think is ideal for tissue culture? A tissue with differentiation capabilities, dedifferentiation capabilities, or redifferentiation capabilities?
All of them.
Yes, any can work. Okay. If it has differentiation capabilities, of course it will differentiate. But if it has dedifferentiation capabilities, that will also work in tissue culture. And if it can, so after dedifferentiation, it has to dedifferentiate. That's the process. Very good. So with this, it comes to an end of vegetative propagation. So I give you all 3-4 minutes. I'll take a quick water break. Okay. You allow me a water break, everyone? Just a quick break. Please allow me a quick break. And in the meanwhile, just go through asexual mode, both vegetative propagation and asexual mode, whatever we have done till now. Is it okay? Is it fine? Just a small break. Yes, I'll come back in 3-4 minutes. Okay, just stay there, and then we will start with the sexual mode of reproduction. So this is where I end the asexual mode. Finish the asexual. Okay, I'll be just back in 2-3 minutes. Okay.
Yes, sir.
sir.
Okay. Done with the revision? Any doubts? Atif, Minashi,
No, sir.
Mohammed. Everyone is back? Okay. So now let's begin with the sexual reproduction part. So which one would you like me to start first? Sexual reproduction in flowering plants or humans? Animal? I think we are discussing plants, so let's stay in the flow and finish with the flowering plants, flowering reproduction in flowering plants first. Is it okay?
Yes, sir.
Yes. So flowering plants. So topic is sexual reproduction. Tell me quickly, what do we mean by sexual reproduction? Just the basics from above. Quick. Sexual reproduction in flowering plants. What do we mean by sexual reproduction? It is where both the sexes are involved, okay? And gametes are formed, so gametogenesis happens, as well as gamete fusion also happens, okay? Correct?
Yes, sir.
Now, when we talk about sexual reproduction in flowering plants, the word flowering plants is very, very important here because its flowers are the reproductive structures of a plant. Right on first thing, flowers. Why are flowers formed? Flowers are reproductive structures. Plant organs. Now, flowers are a very, very amazing evolution that plants did. And if you see basically what are flowers, they are modified. What? Which structure of a normal plant you think got modified and evolved as flowers? So non-flowering plants, plants that do not flower versus plants that do flower. Which part do you think got modified? So there are plants that do not flower. Still, they can undergo fruit formation and things. So plants that do flower, that flower is nothing but a modification of leaves. Is it clear? They are modified leaves. So leaves are at the very end. Root, stem, branches, leaves. Flowers also are at the very end. You will see flowers towards the end of the plant so that it can easily be accessible by birds, insects, honeybees, etc. Because flowers are a way for plants, because plants cannot move, correct? Right?
Yes, sir.
And if plants cannot move, then how are they going to do the exchange of gametes? Tell me, what do you think?
With the help of pollinators.
Yes, they do it through other factors, external factors, which are called pollinators or pollinating agents. Now, these pollinating agents could be animals, could be living, or also can be non-living, like wind and water. Through wind and water, also gametes are exchanged between plants, correct?
Yes, sir.
Yes. Now, these flowers are a way to lure, just attract the pollinators, because flowers, mostly you will see all the flowers, either they have two things: they are very bright in color, so they attract through visual cues, or they have very good scent of smell, right? They have a good scent, they have a good aroma, so they attract through the smell. Or they have nectar, so they also attract through the smell of the nectar, that very delicious thing that honeybees look for, correct? You understand what I'm trying to say? You must have seen honeybees getting into the flowers, going inside and then coming out. right?
Yes, sir.
Yes. So what basically plants here are ensuring is that let's make a structure of a flowering flower here and understand some basic anatomy. Okay. So at the base of every flower, this is the general anatomy. You will see that the flower, any flower is attached to the branch with, so this is where it's attached to the branch. So this is attached with this structure, which is called, what do we call this structure as? Through which we are starting a flower upside down. From down to up, from its attachment to the branch. It's called receptacle. And you all should keep drawing with me, leave some space because we'll reach till here. So receptacle, just like reception, receiving, okay? So reception is which receives everything. So receptacle is which is the first thing that is attached to the, that keeps the flower and the plant together, okay? Receptacle. As we move away from the receptacle, you will see that it starts bulging into this kind of a structure. Can anyone tell me what is this structure? What is this structure? And it then has some green leaves also on all the sides. What do you think is this structure? So what are these green
The leaves are called sepals.
are called?
Sepals.
Yes. So these leaves are called sepals, correct. When the flower is in the bud form, these sepals are closed, correct? It looks green. The bud looks green because it's enclosed in the sepal. So first the sepal opens and then from inside the petal opens. Is it clear? Petal is the colorful part. Right, everyone?
Yes, sir.
This part which is bulging out here actually goes on and forms the ovary. Okay. And inside it are the ovules. So ovary is covered with sepals. This part here is ovary. You will see this. This is the female part. So I am drawing the structure of a bisexual flower. What do I
There's
mean?
also a part called the pedicel, right?
Yes, I'm coming to ped. We'll be coming to pedicel. First, sorry. This board is written. Then you will see a structure going upwards from the ovary. This is called correct. This is the female part of the flower. So what is this structure called? What is this complete structure called? From
Pistil.
yes, the female part is called pistil. If I talk about it here, from this point onwards to the ovary, just above sepals, it's called pistil. It's also known as gynoecium. The word gyne, go back to the terminology section and write down. Who's a gynecologist? Gynecologist. Gynecologist. A doctor. Who do we call a gynecologist? What is gynecology? The word gyne is always associated with female reproductive organ. So write down in your terminology The word gyne is associated with female reproductive system. Is it clear, everyone? Yes or no?
Yes, sir.
Yes.
This part is the pistil, it's the gynoecium, the female part of a flower. At the top, so this is ovary, this is stigma, this part is called style. Stigma, style, and ovary forms the gynoecium or pistil. Is it clear?
Yes, sir.
Above the sepals. Clear to everyone? You also must have seen structures like this growing from this sepal only, which goes all the way and are bilobed at the end. Correct? In a flower.
Yes.
What are these structures called?
Stamen.
They are the male structures. Okay. Okay, when we are talking about female structures, we forgot to mention that in the ovary, which is the female part, what should be present? The female gamete. What is the female gamete known as?
Ovule.
Yes. So inside this ovary, you have a chamber where you can have one or more than one ovules. Okay. So these are ovules. Now this number of ovules will later decide the number of seeds that are present in a fruit. And you all know that there are many fruits which has just one single seed inside, right? For
Yes,
example,
sir.
for example,
Mango now.
mango and
Avocado.
sorry, avocado. Yes. So they have single seeds inside, which means inside the ovary, how many ovules were there?
One.
One that got fertilized. Even if there were multiple ovules, only one got fertilized and only one seed got formed. If it is multi-ovulated, like in a papaya, so papaya is nothing but the ovary. You know what is fruit? Fruit is nothing but the ripened ovary. We basically eat the ovary which is swollen and ripened. That's why when you cut any fruit, inside in the center you will see a little empty space and seeds around it, inside it. So we are actually eating the ovary if it's a true fruit, and inside the ovary the ovules become seeds. So if it is multi-seeded, it means that multiple ovules got fertilized. There were multiple ovules inside. Is it clear, everyone?
Yes.
Yes. Now talking about the male part here, this male part is called anther. It is called anther.
But only the top part is called anther, right?
Sorry?
Only the top part is called anther,
Yes.
as far as I know.
Stamen. Thank you for the correction, Atif. It's called stamen. Now this part, which is this bilobed part, look at these two lobes here. It's called an anther, and anthers could be bilobed or monolobed. So this is a bilobed anther. And what is this part called? Filament, because it's just supporting the anther on the top. Now, if I make the structure of anther in some greater detail here, anther has two lobes in this case, and inside the anther there are small spores like structures which are formed, and these are called
pollen grain.
Yes, pollen grains. Now this pollen grain are the male gametes. Okay, these pollens contains the male nucleus. So just like in humans, there is a sperm and an ovum. Sperm contains the male nucleus, the main genetic material, and ovum contains the female nucleus or the female genetic material. Both fuse together in the process of syngamy during fertilization. In anther also there are pollen grains, and pollen grain can have varied number of structures, but if you see a normal common pollen grain, the most standard, then it has spiny structures at the top with some holes present, inside which there is a cell. So the top covering is just for protection, okay? And there are certain holes. These holes are important because through these holes, the male nucleus which is present will come down, okay? When it will form a tube, a fertilization tube, which we call as the pollen tube, okay? Remember that? I think you must have done this experiment where basically what you do is you let a pollen fertilize on, you let a pollen grow on a stigma and then cut that structure, put it under microscope and look at the growing pollen tubes. Have you done this in your biology class or no?
No.
No. Okay. So we'll discuss that in our classes. Okay, the experiment, because that is important. So this is the male part. This is the female part. These are the reproductive structures. Along with this, there are there are very lavished petals, which are these red. What I'm drawing in red here are petals. Okay, so petals serves only one purpose, and that is attracting the pollinator by color. If they are very bright red, by color, or if they are white, then by scent or the aroma that they have. Is it clear, everyone? I am not drawing. I am removing the petal part for now, but you know where the petals are, right? I'm removing the petal part for some clarity that we are going to discuss. Okay, so this is the major part that we are going to discuss in sexual reproduction in plants. Now write down certain things. Okay, or what I'll do is with the discussions, I will come to the process of fertilization in plants in the next class. Will that work? We will start just with that experiment. So what you can do is go to, before coming tomorrow, go back and look at this experiment that I'm talking about. Type on internet growing pollen tube experiment. Growing pollen tube experiment. You will understand what happens, the how part of this fertilization process that we have to discuss. So we'll take this, the whole fertilization event in the next class. Okay. Clear, everyone? Any doubts? Because it's time for me to leave the class for the Ramzan timings. Any doubts anyone has? No? Okay, then I see you tomorrow, everyone. And come prepared, and we'll discuss the sexual reproduction in plants, and then we'll move to humans. Okay, cool.